We're fortunate to have two incredible speakers with us today, Remco van Soest from SCIEX and Kristina Jurcic from BSI. First, I'd like to introduce Remco, who is an application scientist at SCIEX and brings a wealth of experience in analytical chemistry. Remco studied analytical chemistry at the Vrije Universiteit in the Netherlands and has over 25 years of experience in developing nano and micro HPLC instrumentation and applications in the life sciences. Since joining SCIEX in 2010 through the acquisition of Exogen Technologies, Remco has been focusing on developing LC-MS applications in proteomics. He will be presenting alongside Kristina Jurcic from the BSI team, our product care manager, who will share her own expertise on Glycoproteomics and data analysis using our PEAK's GlycanFinder software. Remco and Kristina will take us through the latest mass spectrometry techniques for glycopeptide analysis, including the innovative use of the ZenoTOF 7600 system with EAD fragmentation and how it enhances the structural characterization of glycopeptides compared to traditional CID methods. They will also explore the power of our PEAK's GlycanFinder software in enabling precise glycan identification and localization, leveraging AI-driven analysis for robust glycopeptide identification. And together, they'll discuss the seamless integration of SCIEX OS and PEAK's GlycanFinder for efficient data processing and how this workflow is transforming glycoproteomic research. Finally, they'll dive into some real-world applications, including human plasma studies, and showcase how over 1,000 N-glycosylated peptides were identified, which will continue to advance our understanding of glycosylation and biological systems. The session will be followed by a 15-minute Q&A where Remco and Kristina will be happy to address any questions you have about these exciting new capabilities. So we ask that you please stay muted for the duration of the webinar and save all questions until the end. But without further ado, I'd like to welcome Remco and Kristina to guide us through these cutting-edge advancements and how they can enhance your research.
We certainly have a special guest, as Braden mentioned, Remco van Soest. I'm not sure how I'm pronouncing that, but happy to welcome him. He is our colleague at SCIEX, who will be talking about some of his experiments done using ZenoTOF 7600 system, specifically for glycopeptide analysis. And most of you are likely familiar. ZenoTOF may use CID for MS2 fragmentation. However, there is a unique fragmentation mode called electron activated dissociation fragmentation, or EAD, which is particularly useful for looking at labile PTMs such as glycosylation. So I will let Remco talk more about this, and I'm going to start basically with some of the introduction and characterization of our GlycanFinder software for glycoproteomics analysis. So first, I'll talk a little bit just about the glycoproteomics and some challenges, and then I'll talk more about our GlycanFinder, our BSI comprehensive solution for glycoproteomics, with some details, of course, on the software features, how the scoring is delivered, and how the result is then provided. So to start off, we know that protein glycosylation is one of the most prevalent, complex, sophisticated post-translational modifications and has a large impact on many biological processes. Essentially, it's a simple, the attachment of the carbohydrates of diverse structures to different sites on the protein. And essentially, these attached glycans may promote stability, folding changes of the proteins, and play an important role in regulating, of course, these proteins' functions. So dysregulation of glycosylation may be associated with various diseases. The aim, of course, of glycoproteomics is kind of to provide a detailed characterization of the glycosylation events in biological samples. However, the actual accurate identification of intact glycopeptides remains quite challenging. One of the things is high glycan complexity, meaning we may have a covalent attachment of a single sugar unit, multi-sugar polysaccharides, or complex oligosaccharides. In addition, they can be N-linked, they can be O-linked. And another thing is that we have the presence of both the glycan and the peptide fragments. So these make the mass spectra and the annotation much more complicated, as we need to decipher both the peptide backbone as well as the actual glycan composition and the structure in our case. And last, it's essential to try at least to address the heterogeneity problems, that there may be multiple glycosylation sites per peptide, and also multiple structures with the same glycan composition or the mass. So GlycanFinder kind of tries to address these challenges in terms of the main objectives of glycoproteomics. So identifying glycopeptides and then glycoproteins is the goal, providing glycosylation site profiling, as well as glycans profiling. And here's our paper, actually, that we published on GlycanFinder, if you wish to check, back in 2023 with some more details. But essentially in this paper, we actually kind of explain our algorithm logic. So I'm going to try to start with this, just to give you a bit more idea. So the first thing we do is we identify a glycopeptide, excuse me, MS2 from oxonium ions. So the presence of the oxonium ions will indicate a glycopeptide spectrum. Next, we perform a peptide-based search. So what this is basically a classical matching of the peptide backbone to the protein database, and then the glycan fragments against our built-in or user-defined glycan databases. If we have a good match, then we basically move these glycopeptides forward and we apply, say, a 1% peptide FDR control. Now, the rest of the MS2 spectra are moved, so those which are not, we did not find a good peptide and a glycan, are moved forward to what we call a glycan-based search. So here, typically, we have poor peptide fragmentation. And if a good glycan is deciphered, because oftentimes your glycan fragments are quite intense, or more easily actually acquired, then what we do is we go into this pool of peptides now and we check, basically in this pool, we know what was confidently identified. So we match and align the found glycans here to the peptide sequences confirmed, and basically we then move everything forward, combine this result with the added glycopeptides, and we apply a glycan FDR control, so the final control. In case you want to move forward and look for some new glycans, we also have a function to do some glycan de novo sequencing. Now, as I said, the FDR control here includes both the peptide and the glycan controls. Again, the peptide FDR is fairly straightforward. We use the decoy protein database to kind of estimate that peptide FDR from the decoy peptides and the target peptides. Now, for the glycan FDR, we need to create these decoy MS2 spectra. To do so, we kind of apply mass shifts of, say, 3 to, I think, 30 m over z to all of the fragment ions in our MS2 spectra, and we create these decoy spectra, and we use them basically to do the ratio to calculate our final glycan FDR. And again, just to verify, the glycopeptide will pass the control, so let's say 1% FDR control, only if both the peptide and the glycan FDRs are satisfied or less than that 1%. So that was a quick kind of look at the algorithm. And I'd just like to kind of list the key features of the current GlycanFinder here. And I actually like to mention that in 2 weeks, we are releasing a new GlycanFinder, our new upgrade. So keep tuned. We'll talk about those features soon. For the current one, we have both N-linked and O-linked glycan profiling with structural resolution. We have panorama profiling of glycosylation sites with multiple enzyme digests. This is kind of similar to our PEAKS-AB, where we overlay different digests to give you a higher confidence in the glycosylation sites. Glycopeptide quantitative analysis is available, label-free labeling methods, typically chemical, such as TMT. The Novo sequencing, as I mentioned. We have our Glycan database viewer that I'm going to show. And I'm hoping that the graphical user interface is intuitive, easy for validation and reporting or your kind of needs. And we are vendor neutral. So just at a glance, these are some of the key featured kind of results that GlycanFinder provides. This is the glycan protein coverage view, very similar to our protein view in Studio. We have glycan site profiling showing some abundances of different glycans for a specific site. And we can also do some glycans profiling across your sample set. So for each of the glycan structure, we can show you the abundances across your sample set or experiment. Of course, one of the key things for us is to provide the user with some confidence scores for the glycan localization, but also for the confidence in the structure of the glycan reported. So I'm going to start with the site classification. Essentially, we can have different scenarios. Glycan site can be very easily determined directly. We see all the necessary ions. In some cases, there's only one possible site, so that's easy. In other cases, we may be able to decipher or exclude one, but not all of the sites, so we have still some ambiguity. And in some cases, of course, it's not possible, given the MS2 spectra, to determine the exact site of glycosylation. Either way, we use our standard scoring, our classical scoring for the site localization, which is an A score. Those who use our PEAK Studio or PEAKS Online will be familiar with this score. Essentially, it is a -10 log P value, where the P value is the probability that the modification occurs at the reported position compared to other possible positions, meaning that a -10 log P, say, of 20 would be a P value of 0.01, which would be 1% chance that this is actually a random assignment to this site and not a confident site. So 1,000 is obviously very high and pretty much what we report as a maximum. I mentioned that we have built-in glycan databases. So our glycan databases contain, and they require, if new glycans are added by the user, structural information. This means we need all the possible ions for each of the compositions or structures, which can then be used to derive that glycan structure. And having said that, each of our glycans or glycopeptides will have a glycan score calculated depending on the glycan fragments basically found in the spectra. Now, of course, it is possible to have multiple glycans that have same composition, same mass, but different structures calculated. And what we do is we, again, give them the scores, and we then take the top glycopeptide and we calculate the normalized score gap, or the difference between basically the top glycan score and the second top glycan score. And this is expressed as an S score in percentages, and it basically tells you about the structural resolution. So S score reflects the confidence in the structure, glycan structure assignment, while the A score reflects the confidence of the glycosylation site localization. We can also perform some de novo sequencing to find or identify potentially new glycans, and these glycans can then easily be added into the database. For the de novo sequencing, we use deep learning approach to construct, this is over my head for sure, so I'm just going to simplify it and say our model constructs basically a glycan tree by iteratively predicting and adding monosaccharides to the tree. And this is done for both N and O-linked glycans. And one last thing that is kind of helpful, talking about this de novo sequencing, in our glycan database viewer, which is essentially an editor, you can pick any of the already structures existing, or you can create a new one, but you can easily edit and add any glycan needed. So this way you can easily customize the database. It is also possible to add glycans in a text format directly into configuration. It has to be a glycoCT or GDP structural format though. And just to finish off, I would like to mention a very recent publication, 2025, by Dr. Liang Chao, who used ZenoTOF mass spectrometer with a dynamic CE-CID, basically and then GlycanFinder to identify glycoproteome profiles from cancer and control groups. In fact, they truly emphasize the importance of glycoproteome research when showing that much more difference in these diseased tissues or cell lines were found, in fact, when looking at glycoproteome compared to a proteome. Now, they further compared in their studies our GlycanFinder, our Studio, like our general kind of proteomic solution with the Glycan search, and FragPipe. And you can see that FragPipe definitely produced the highest number of glycopeptides. However, I do want to note that FragPipe is a compositional search only. So along with perhaps MaxQuant, Bionic, they are all compositional search. While GlycanFinder is a structural search, meaning it has another level of filtering for hopefully another level of confidence. And in fact, in our publication of the GlycanFinder, we do show kind of similar behavior in terms of, again, FragPipe producing higher numbers of these compositions of the glycans. However, we went ahead and we compared our GlycanFinder to a structural, so another very well-known and I think very good structural software for glycoproteomics, pGlyco3 or PG3, and you can see that the numbers are actually fairly similar and somewhat the overlap. And this is just a last figure kind of shown from Dr. Chao's paper, not to go into any detail, but to again reemphasize the power of ZenoTOF for both proteome and glycoproteome especially, or really that glycopeptide fragmentation, acquisition, and then the GlycanFinder to interpret these spectra and identify and profile the glycopeptides. And with this, I would like to thank you for your attention. I'm going to pass my screen on to our dear guest, Remco, who can give you some more detail on the EAD fragmentation unique to SCIEX technologies, which achieves increased sensitivity and confidence in glycopeptide identifications.
Okay, thanks, Kristina, for inviting me at this webinar. So you gave a great overview of the software. I could learn even myself quite a bit of this. But I will continue with giving just an example or sharing some of our experience using the GlycanFinder software on data that was acquired on the ZenoTOF 7600 system. So this is briefly the outline of what I wanted to cover. I will give you a really brief overview of the ZenoTOF 7600 system, specifically focusing on the features that are important for glycopeptide analysis. Talk a little bit about sample preparation, because the one thing I learned is that is really critical to be successful in glycopeptide analysis. Talk a little bit about the optimization, and then share some results and end with a conclusion. So the ZenoTOF 7600, meanwhile 7600 Plus, it was launched in 2021. And then it was updated last year with some additional scan functions, which we won't discuss because they are not applicable to glycopeptides at this point. So there's two features in the ZenoTOF system that are very important for glycopeptide analysis, right? So one of them is the ZenoTrap, which I will explain a little bit more in detail over the next couple of slides. And then, as Kristina mentioned, the other one is the ability to do electron-activated dissociation rather than CID, so collision-induced dissociation. So before I start talking about the ZenoTrap, again, keep in mind that glycopeptides, the main issue is that they're very low abundant, right? So glycosylation, not every protein, well, many proteins are glycosylated, but not every protein is. And then also the proteins that can potentially be glycosylated, they're not completely glycosylated, right? So when you try to identify glycoproteins by looking at glycopeptides, you're typically dealing with pretty low abundant glycopeptides. And then, as Kristina showed as well, there's many versions of glycosylation, right? So you may have many different glycans on a specific glycopeptide, right? So that all causes that these glycopeptides are pretty low in abundance, right? So you need a very sensitive mass spectrometer, right? So the ZenoTrap will give you more sensitivity. How does that work? So typically on a quadruple time of flight instrument, the cycle time, the duty cycle is not very high. What does that mean? It means that you're only analyzing about 5 to 25% of the ions. And the reason for that is the ions are transferred from the quadrupole region kind of in a constant fashion, right? So there's always ions after fragmentation coming from the quadrupole region going into the TOF accelerator region. When they transfer, as you can kind of see here, there's already some kind of a little bit of separation, unfortunately, right? So you're basically sampling not all the ions of a specific M/Z value that you potentially could, right? So then they're extracted, but while you're doing the TOF analysis, the quadrupole is the ions going to the quadrupole are just continuing. So many of the ions are basically lost for the analysis, right? So that explains the relatively low duty cycle, and therefore which will affect the sensitivity, right? So what did we do with the ZenoTrap? We added the trap after the fragmentation area in the quadrupole region. So it effectively traps the fragments, right, if you're doing MS-MS. And then it moves the fragments in a more coordinated fashion to the entrance point of the TOF accelerator, right? You see now they're all kind of concentrated together, right? So they're released with a different time scale, so they all arrive at the TOF accelerator at the same time. Then we eject them, right? And what's not really shown on this animation is while you're doing that, you're already collecting the next set of fragments in the ZenoTrap, right? So that effectively makes that you don't lose any ions, and therefore it increases the sensitivity quite a bit. So we typically see sensitivity improvements between trapping, so using the ZenoTrap or not using the ZenoTrap, in the order of around 10 times or so, right? So quite significant. Again, very important for glycopeptide analysis. So the other feature is as Kristina mentioned, is the electron activated dissociation. So that works by generating electrons. They are then captured by the ions, and then they form an intermediate radical state, which then falls apart into fragments, right? So it's kind of like ECD, but with the additional advantage that you can tune the fragmentation, right? So you can set the kinetic energy for the electrons that you use to do the fragmentation in the EAD cell. If you use a very low kinetic energy you can do that for multiply charged peptides and proteins, maybe for middle down sequencing. We've had many examples of that. But then what we will use, we will add a little bit more kinetic energy, and then you can use that for fractionating glycopeptides, but not just glycopeptides, also phosphorylated peptides, disulfide bonded peptides, etc. Right? So why do we do that? What are the differences with CID? Well, with CID, for example, if you talk about glycopeptides, mostly what you will see is most of the energy will go into breaking the glycan off the peptide and breaking the glycan up in pieces, right? So you don't really get fragments that will tell you the sequence of that particular glycopeptide, right? So that will hinder you. Yes, you can identify if a a peptide is a glycopeptide or not, but then you don't really necessarily know where it was glycosylated, and you also can't often not figure out the sequence, right? So with EAD, it's a very different fragmentation. It also generates different types of ions, right? So with CID, it's mostly B and Y ions, and with EAD, you create mostly C and Z ions of the peptide backbone. But it also can maintain the glycan on the peptide, right? So that will help you in identifying where was the glycosylation, or where is the glycosylation, and it will also help you in actually getting information about the glycan itself. Before I go into the showing you some of the data, again, we're only going to talk about N-linked glycosylation at this point, right? So glycosylation can be N-linked or O-linked, so N-linked on asparagine. That's what we're doing right now, mostly because the sample preparation that you do will have to be kind of targeted to either N-linked or O-linked glycosylation. So maybe at future stage we could do something with O-linked glycosylation as well. So again, this slide nicely shows again, the difference between CID and EAD for the fragmentation. So CID, it basically breaks off the glycan, breaks the glycan in pieces, and you'll end up with these low M/Z value ions here in your MS spectrum. These are oxonium ions that again can indicate that they came from a glycosylated peptide, but you have very few B and Y ions that will help you in identifying what the peptide structure, what the peptide sequence was, and also where the glycosylation took place, right? So with EAD, that's very different. I'll show you later in some real examples as well. But now you actually maintain the glycan on the peptide fragment, so you will see lots of fragments with the glycan intact. You will also get a nice series, typically of C and Z ions, that will help you in identifying the sequence of the peptide. So the workflow that we used is, like I said, one of the issues, even with the ZenoTOF system that has the Zeno trap that will help to improve the sensitivity, there's still very low abundant peptides typically, right? So, and also, if you try to do this in plasma, as most of you, of course know, is that there's 14 proteins in plasma that make up, like most of more than 80 or 85% of the entire protein content of plasma, right? So that's not helping with the analysis, right? So what we did is we depleted the plasma. We used the immunoaffinity based depletion column. We did a digestion, and then we followed the procedure to enrich the tryptic peptides for glycopeptides, specifically using a helic resin. I'll show a little bit more detail of that in the next slide. So as far as chromatography goes, again, because we do need to get all the sensitivity that we can have, right? We use nano HPLC, trap-and-elute nano LC. I like trap-and-elute because it gives some additional cleanup step. It prevents the nano column from being prematurely getting clogged, and it also allows you to vary the injected volume very easily, right? We use a 60-minute gradient, and we use a combination of a Phenomenex nano LC column with a Waters nano LC trap. So data acquisition, I will also talk a little bit more later what's important there, but we used either EAD or CID based MS in a data dependent setup, right? So more about that later. And then Kristina talked about the data analysis. We use the GlycanFinder software for that. And then I also show that you can also, on the same data, on the TOF information, you can do quantitation of the identified glycopeptides as well, using our own quantitative software analytics. So on the sample preparation, so we started out with 100 microliter plasma, depleted. So then most of the proteins are gone, right? So that will help you again with identifying lower abundant glycoproteins. We did a digestion, typical kind of fairly standard digestion, but on a filter, so filter-aided protocol. And then we did the enrichment. So I kind of was in the fortunate position to have a colleague here who spent most of his PhD time on developing methods for glycopeptide enrichment. So he showed me how to make these little spin columns from Eppendorf tubes and a pipette tip. You pack them with a mixture of cotton and hydrophilic stationary phase. So the cotton actually has a very good affinity for specifically glycosylated peptides. It acts kind of like a helic phase, a very cheap helic phase, of course, and it also holds the helic beads together in the pipette tip, right? So that provided, as you will see later, a very efficient way of enriching your sample for specifically glycopeptides. So on the mass spec method side, I won't go through all the details, but it's a data dependent acquisition method, right? So what that means is the mass spectrometer on the fly will look at the TOF mass spectrum. It will look at the most intense precursors, and it will do MS on those, right? So one of the important parameters that you can set in the DDA method is the number of precursor candidates, right? So how many of the precursors in a given cycle do you want to do MS on, right? So at first thought, you would think, well, you want to make that as high as possible, right? So that's what we did initially. So we used a 50 millisecond accumulation time for the EAD fragmentation. And then we set the max candidates at 36, right? So that generated a lot of spectra, but only a fraction of those were actually spectra from glycopeptides, right? And we identified around 500 glycopeptides. So we played a little bit with that, keeping the cycle time constant, but varying the accumulation time for the EAD experiment. And then, of course, we had to lower the number of candidates, right, if you want to keep the cycle time constant. So interesting for us to see was that, yeah, you obviously will reduce the number of MS spectra, but the fraction of those that are actually coming from glycopeptides or could be identified as coming from glycopeptides was a lot higher, right? So therefore, we identified also more glycopeptides. So we moved further with this particular setting. So relatively low number of candidates, but with a high, relatively high fragmentation time or accumulation time for the EAD fragmentation, right? Because EAD, it gives you lots of information, but it is generally a little bit less efficient compared to CID, right? So you may end up with lower sensitivity compared to CID. So this helps with that, of course. So here's just an example of the tick from the TOF and the tick from the EAD MS experiment, right? And in this first test, what we wanted to do, we wanted to see if our enrichment actually worked, right? So this is a comparison of just the depleted plasma, right? Equivalent of half a microgram. And then here, in order to maintain approximately the same kind of intensity, we had to take a lot more, right? So this was the equivalent of 37.5 microgram of plasma protein, right? So obviously during the enrichment, you lose a lot of the non-glycosylated peptides. So then we used the software, the GlycanFinder software, and we looked at, okay, what's the fraction of glycosylated peptides versus the total number of peptides. And as you can see very nicely here on the left side, you see the enriched depleted plasma, and you see the majority, pretty much more than 90% of the peptides are indeed glycosylated peptides, right? Whereas in the just depleted plasma, it's exactly the other way around, right? So this enrichment really worked very effectively and really helps you to identify more glycopeptides. So here's the first example of using the GlycanFinder software. So a couple of things that I had to get used to is the spectra are processed, right? So they're not the raw spectra. I'll give you an example of a raw spectra in the next slide that was underlying for one of the identifications in this spectrum. So they're de-isotoped and decharged to a +1 charge state, right? That's why it extends all the way to almost 4,000 here. But like I said before, what you can very nicely see already is, you see the very nice C and Z series of ions here that helps you in identifying where the glycosylation took place. And also it helps you in sequencing the entire structure very nicely, right? And then here you see the fragments with the glycan still intact that will help you to confirm the position, but also helps you to confirm the structure of the glycan itself, right? So this was a relatively high concentrated protein, so therefore glycopeptide. But oh, sorry, I was going to show you this as well, right? So this is the underlying the spectrum, so the raw data, which you can also always go back to in the GlycanFinder software. I found that rather nicely. And you can see that for this particular C9 fragment, you can see where the information actually came from, right, before the deconvolation. Okay, so this is a much lower concentrated glycoprotein, right? So peptides from a glycoprotein, Glycan protein. And here we kind of also acquired data using CID. And like I explained before, you can see the difference quite nicely, right? So here with EAD, you see a nice series of C and Z ions. You see the peptide glycan fragments here. With CID, you get the oxonium ions that will again indicate that it's a glycopeptide, but doesn't tell you too much about the structure itself, right? So this is kind of summarizing all the data that we got on this particular application. So we run it in triplicate. It's a DDA experiment, right? So you will never see exactly the same glycopeptides in each run, right? So that's why you typically run it multiple times, and then you kind of sum the results for the 3 runs. So in those 3 runs together, we found just a little over 1,000 glycopeptides, which, when I looked into the literature, it's a pretty good number, right? And these were coming from 98 proteins, right? So the other thing that we looked at is quantitation. So you can actually do quantitation based on the TOF MS data in the GlycanFinder software as well. But we have very nice software for quantitation ourselves that we use, mostly for quantitation by looking at fragments. But in this case, since it's a DDA experiment, you can't do that, but you can do quantitation on the TOF MS data. And the quantitative software that we have, it's called Analytics in SCIEX, can be used for that as well. So what I did here is I just exported all the monoisotopic MOVZ values for all the identified glycopeptides, exported that from GlycanFinder, imported in SCIEX, and did quantitation on that, right? So doing that, I got this CV of less than 20% for more than 90% of all these identified glycopeptides, which was pretty good. I think just considering it's just on the TOF MS data, right? What actually you can also do for the somewhat more lower abundant glycopeptides, you can sum isotopes, right? So you can set up the quantitation software to do that for you. Once you have that done in the method, it will do it automatically on every sample for you. And as you can see here, it very nicely increases the signal to noise ratio, right? By just summing the multiple isotopes from a glycopeptide. So before I stop, I just want to mention this. It's the position of the EAD cell is before the CIDs, the Q2 where you can do CID in, right? So that has prompted people to try a combination of EAD and CID. So the idea is to first kind of sort of activate the precursor, and then maybe not completely, maybe get EAD fragments and then do some additional CID to get some oxonium ions, right? So the idea is to get a hybrid spectrum that will give you both CID type fragments and EAD type fragments, right? So there's 2 groups that are actively working on this using the ZenoTOF 7600. One is Albert Heck's group in the Netherlands. He's got some nice data on that, and he kind of called it EAD CID. And then there's a lab in Australia as well, Benjamin Schultz's lab at University of Queensland, that has also published about that, right? So that can potentially increase the number of identifications of glycopeptides even further, right? Currently, you could not process data like that in GlycanFinder, but in the next version, the sort of 2.5 version, that will be an option as well. So I'm going to work on that together with Kristina and see if we can improve the number of identified glycopeptides even further using this hybrid fragmentation. Yeah, that's kind of what I wanted to talk about. In conclusion, I think I've showed that the EAD spectra are very different from CID spectra. They give you a lot more information for glycopeptides, so you can confirm where the glycosylation took place. You can get a better sense of what the glycan structure was. In this particular example, where we depleted and digested genome and plasma sample, we could find over 1,000 glycopeptides. And then the SCIEX OS software allows you also to do quantitation very easily on the same data set using the TOF MS data.